Yay. Okay, wait, we're almost there.
Super.
So
your slide is up now.
Is it the whole slide that's filling the screen?
There, no, your screen is divided and your slides are on the left, and your first slide is taking up 2/3 of the screen on the right.
Okay, how about that?
That's correct now. You
Yay.
ready?
Now I'm going to hang up.
Okay, I hope we don't lose you in the intro.
God forbid, we'll never get it back. Okay, thanks, Steve. Bye.
All right. Well, welcome everyone to the Portuguese Water Dog Club webinar on chondrodystrophy in the Portuguese water dog. I'm pleased to introduce our speaker, Dr. Danica Bannasch. Dr. Bannasch is a veterinary geneticist who specializes in canine genetics. She obtained her PhD in molecular biology from Princeton University and her Doctor of Veterinary Medicine from the University of California at Davis. She has been a professor at the School of Veterinary Medicine at the University of California, Davis since 2001. She was appointed the Maxine Adler Endowed Chair in Genetics in 2013. And in 2019, Dr. Bannasch was awarded the International Canine Health Award for her research accomplishments in canine genetics. Dr. Bannasch has published over 100 peer-reviewed manuscripts, and her laboratory has identified the causative variant for 16 inherited diseases in dogs and horses. She has collaborated on the identification of many more. Her research goals are to improve the lives of dogs through understanding the relationship between their genetic variations and the resulting phenotypes. In her spare time, Dr. Bannasch breeds dogs and competes in conformation, agility, hunt tests, tracking, and barn hunt with her Nova Scotia duck-tolling retrievers and her Danish Swedish farm dogs. Please post any questions you may have in the chat box during the presentation, and hopefully Dr. Bannasch can get to many of them during the Q&A, which I will monitor. And here now is Dr. Danica Bannasch.
Thank you so much for that introduction. I am so sorry about all the technical difficulties. Apparently, even though we tested everything out, Microsoft Teams doesn't work very well with UC Davis somehow. So I'm at a little bit of a disadvantage because I can't see any of you, but hopefully you can see me and see my slides. And at the end of the presentation, I'll probably click off of the slides to answer your questions. So I do have a little association with Portuguese water dogs because my parents had a couple of Portuguese water dogs for years. So you'll see some pictures of their dogs scattered throughout and pictures of a friend of mine's Portuguese water dogs. Okay, so we will get started. And if hopefully someone will interrupt me if you no longer can see my slides. So I wanted to start off just with a definition, and that is the term chondrodystrophy. So chondro means related to cartilage or bone, and dystrophy means tissue degeneration. And chondrodystrophy was a name coined for this disorder many years before I ever started working on it. And this tissue degeneration aspect is something that we're going to talk more about. At UC Davis, we hold dachshund races every year, which always makes me cringe. So far, there hasn't been a herniation event that's happened during the races, but it seems like a really bad idea to me. So,
Dr. Bannasch, can you try to center your slides? We're seeing about 2/3 of the slide with them being cut off.
Oh, I do not know how I could possibly do that because I have so little control in this mode.
Perhaps if you leave your upcoming slides on the left, we can see the whole slide in this view.
So in this view right now, can you see the whole slide?
We can, as well as your upcoming slides, but I think this is preferable.
Okay, hold on. Let me just make it as big as I can, the actual slide. So
Okay,
does that make it? Go ahead. Better or worse?
That's better.
Okay. All right. Sorry about that. Like I said, doesn't like my computer. We're a Zoom faculty here. So in 1952, Hansen, who is a Swedish anatomist, did this really amazing and thorough study of the pathology and anatomy of dogs that had disc degeneration, and the breeds that he did are shown along the top here. So he looked at all of the intervertebral discs in these dogs and identified that at a very early age they were degenerating, and so that's kind of where this term chondrodystrophy came in. And later, Brond, who is referenced at the bottom, can you guys see my pointer, by the way? If I like wiggle this around? No?
I'm not seeing it.
Okay. Beagles were later added by Brond, who did the same type of analysis. And Hansen was also credited with defining Hansen type 1 disc disease, which is the type of disc disease that chondrodystrophic dogs get. And I'll tell you, when you look back at this paper, and one of the things that I was initially really frustrated by is the fact that some breeds aren't specifically mentioned, like he lumps all spaniels together. And as we know more about chondrodystrophy, it's actually become apparent why that was at the time, but it seemed really odd to me that you would lump them all together. Okay, now hopefully when I advance, you guys will see the slides advance. So we got interested in chondrodystrophy because we were looking at a phenotype in Nova Scotia duck-tolling retrievers. And as you heard in the introduction, I breed Nova Scotia duck-tolling retrievers. And what we saw is that there were lots of dogs with very sort of short and crooked legs, kind of medium-sized dogs in the middle here, which were kind of the ideal, and then extra long-legged dogs like on the right that were sort of off-type. And so this segregation seemed to be occurring in the breed, and it appeared to be a Mendelian factor in that if you bred the dogs in the middle to each other, you often got dogs that were short-legged and dogs that had too much leg. And breeders were particularly concerned about something we called skeletal dysplasia, which were these tollers that had really short and crooked legs. And so we set about taking radiographs of a lot of Nova Scotia duck-tolling retrievers, and what we found is that there were sort of a gradation. So on the top of this slide are radiographs of sort of what we would think of as a normal toller, in the middle one that's sort of mildly affected, and the bottom one that's more severely affected. And eventually I convinced a graduate student to work on this, and we decided to try and map it. And because there was this sort of gradation of phenotype, we took the two extremes. So we ended up using the dogs that looked like they had very straight normal legs and contrasting those to dogs with these sort of short and thickened bones. And we did something that we call a genome-wide association. So this is where, sorry, I don't know why my slides are looking so ugly, but I guess it's because it's not in presenter mode, so I apologize about that. We did something called a genome-wide association, which is where we take a set of markers across the genome and we look for a difference between cases and controls at the genotypes for those markers. And so when we did this with this form of skeletal dysplasia, we got an association on canine chromosome 12. So this is something called a Manhattan plot, because it's supposed to look like the Manhattan skyline, and the red line shows which markers are significantly associated. So you can see that there were a bunch of markers on chromosome 12 that were associated with this short stature. It's one of the advantages of using breeds like Nova Scotia duck-tolling retrievers is that you don't have to use very many cases and controls to get a significant association. And one of the disadvantages of using Nova Scotia duck-tolling retrievers is that you end up with an associated region which is really large. So this red bar shows where along chromosome 12 the association for skeletal dysplasia and tollers was. And it's a really big region with lots and lots and lots of genes. So we thought, well, maybe we can use other breeds of dogs to narrow this down, because there's obviously dogs with lots of different length legs, and so maybe something about this region would be shared with other dogs. And when we did that, we identified that there was a shared region on chromosome 12. So what I've plotted here is, you could basically consider it sort of the diversity of alleles, and you can see that there's a lot of diversity, and then this region you get no diversity, and then you go back and you get a lot of diversity. And what was going on here is that there was a region shared between certain dog breeds and Nova Scotia duck-tolling retrievers where there was no diversity in those dog breeds. And the dog breeds are the same breeds that had been identified by Hansen as being chondrodystrophic. So I had a really good colleague who's a neurologist, and we had been talking a lot about chondrodystrophy and wondering what was predisposing dogs to disc herniation. And so this was a sort of easy leap for us to say, oh my gosh, you know, these are chondrodystrophic dogs and they're sharing the same region of the genome. We went on to look at chondrodystrophic dogs compared to normal dogs and got the same region of the chromosome. And then we actually looked at dogs that had Hansen type 1 disc disease and compared those to dogs that hadn't herniated and found the same region of the chromosome. At the same time that Emily Brown, who was a graduate student in my lab, was doing this, she also looked through the literature and identified that this region on chromosome 12 had been identified previously. So it was identified by Elaine Ostrander and Gordon Clark, and they were looking at all of the lovely phenotype data from Portuguese Water Dogs, and they had defined that there was a region of the genome that was associated with shorter legs and longer legs in Porties, and it wasn't the same region on chromosome 12. This same region was also associated in a genome-wide association study in dachshunds with disc calcification. So both of these results were really consistent with what we found in the tollers. And I just wanted to show you a picture of these. This is actually a picture of Portuguese dog legs from the paper. And these are the Nova Scotia duck-tolling retriever legs that I showed you. So these are the forelimbs you can see in this picture. These are the feet, the sort of wrist area of a forelimb, and then the radius and ulna. And the phenotype is similar. It's not necessarily super specific, but you can see that in some of the Porties, that dogs have shorter and thicker legs, which is kind of the same thing that we saw in Nova Scotia duck-tolling retrievers. So this is so irritating that it's so ugly. I apologize. I'm just gonna get rid of that because I can't stand it. So we identified that there was an FGF4 retrogene, and I'm going to tell you more about what retrogenes are. But one of the reasons why we were really excited when we made this discovery is that we did know something about dwarfism in dogs before we started this, and that was that there was an FGF4 retrogene on chromosome 18 that was associated with disproportionate dwarfism in dogs. And so this was work actually also done by Elaine Ostrander's group, and they used sort of breed standard definition. So any sort of breeds that were known to have long backs and short legs were counted as chondrodysplastic. And then they compared those to breeds with normal long legs. And they did a genome-wide association and got an association to chromosome 18. Actually, the second highest association that they got in this genome-wide association was to chromosome 12, which was the later on, which we identified as the chondrodystrophy locus. So one thing that was probably very fortuitous is that they used a word called chondrodysplasia, which didn't really have a veterinary definition for it. It basically just means abnormal bone or cartilage, and in this case, what they meant by that was abnormally short legs. Despite the fact that it's a different word than chondrodystrophy, littered throughout veterinary literature, and every veterinarian that you talk to is confusion about what chondrodystrophy is versus chondrodysplasia. And I'll try and clarify that a little bit more here as we go along. But it's not surprising that breeders might be confused by it because veterinarians are confused by it. Okay, so one thing when this paper came out that the neurologist and I talked about is that while there's no doubt that this is associated with short stature or short legs in dogs, it didn't really explain the breeds that were predisposed to disc herniation. And we knew that going in, and this is just an example of that here. Scottish Terriers and Cairn Terriers and Glen of Amal Terriers are not predisposed to disc herniation, but they do have the chondrodysplasia mutation. So this is just an example of how things were a little bit different. Now you guys are not going to be able to see all the cool graphics that I did, which is really sad. Anyway, so these mutations are, try and get rid of this so it's at least a little bit less busy. These mutations are a little bit different. So often, you know, if you have other mutations in Portuguese water dogs, I know PRCD exists, progressive retinal atrophy, and that's where there's a gene and there's a normal version of the gene and then a mutated version of the gene, right? And we run a DNA test to tell you whether your dog has one copy of the mutated version of the gene. Retrogenes are entirely different. So a retrogene is where there's an extra copy of an entire gene inserted in a new location. So they're really kind of exciting biologically, not so exciting if you're a dog that has them. And the thing that we discovered on canine chromosome 12 is that there is an FGF4, and I don't think I can get this to proceed with the animation, but anyway, there's an FGF4 copy that's been inserted on chromosome 12. So the normal chromosome doesn't have an FGF4 gene there, and the chondrodystrophy mutation is that there's an FGF4 gene inserted on the chromosome. So there is no sort of, the normal version is just absence of this extra copy. The weird thing is that FGF4 normally lives on canine chromosome 18. So chondrodystrophy is caused by an FGF4 copy on canine chromosome 12, and chondrodysplasia is caused by an extra copy of FGF4 actually on canine chromosome 18. So that might have led to some confusion in the past. So what does this mean to have an extra copy of a gene? Like, what happens when there's an extra copy of the gene? So Dr. Ostrander's group showed that in dogs with chondrodysplasia, they had increased expression of FGF4. So there was more RNA made from the FGF4 protein. And we did, and I'll show you some studies where we looked at FGF4 expression from chromosome 12 and also identified that there was increased expression. So let me just tell you a little bit about FGF. It stands for fibroblast growth factor, and it's a growth factor ligand that binds to a receptor, and that receptor signaling leads to normal long bone growth. The interesting thing and the sort of relationship between these canine dwarfism genes and humans is that in humans, normal achondroplasia dwarfism is caused by a mutation in the receptor for FGF4. So human dwarfism is caused by a mutation in the receptor. Canine dwarfism appears to be caused by, or a couple of forms appear to be caused by overexpression of the ligand that binds the receptor, the growth factor. This is some data from mice showing where FGF4 is normally expressed. So this is a, I realize you can't see my pointer, but anyway, on the right, on the top is a mouse embryo. And the area that's pink and purple is showing where normal FGF4 is expressed. So it's expressed in the vertebral bottom column as it develops. And it's also expressed in the limb buds as they develop. And this special little purple crescent structure is called the apical ectodermal ridge, and that's where limb growth is initiated. FGF4 is also actually expressed in the embryo at the area where the external ears would develop. And it is not expressed at all in adult tissues. So it's completely turned off in adult tissues, and it should be. So what we found with chondrodystrophy, so this is the FGF4 retrogene on chromosome 12, is that in puppies, we looked at intervertebral discs and found that in those intervertebral discs, there was 20 times as much FGF4 expression as in dogs without chondrodystrophy. So extra copies of FGF4 means that you have higher and abnormal expression of FGF4. So because there's increased expression, and it's probably being expressed at the wrong time and certainly in the wrong place, we call these variants that increase expression gain of function variants, and they are usually dominantly inherited. So if you think about it, if you have one copy of a normal chromosome that doesn't have the FGF4 retrogene inserted, and the other chromosome has a copy of the FGF4 retrogene inserted, then there will be increased expression. And so that would lead to a dominant phenotype. You would see a phenotype because it has a gain of function and extra expression. Okay, okay, so now we'll start talking about disc disease. So I don't know if I have my model in my office. I have a little model of vertebral bodies that I should have gotten out. But so this is the spinal cord, which runs inside the vertebral bodies. And each of these vertebrae is cushioned from the next one by this intervertebral disc, which is shown here in pink. And if you kind of slice it in half, the disc has a gelatinous core that's which is called the nucleus pulposus, which is surrounded by the annulus fibrosus, which is sort of a harder ring around the outside. So what is the purpose of all of these discs? Well, they cushion the spinal cord during movement. So we kind of think about dog skeleton sort of as being really static and just kind of sitting there and not moving. But when you see what a dog skeleton looks like when a dog is running or curled up in a ball, you can imagine that there's a lot of flexibility here. And so these discs are set between the vertebral bodies to allow this sort of cushioning activity. This is, sorry, a picture of a normal intervertebral disc. That's sort of the spine has been sliced in half so that you can see the nucleus pulposus. And you can see it's gelatinous, and you can imagine that it probably provides some cushion. This is the annulus fibrosus on the outside of it, which is a little bit of a harder structure that kind of keeps that cushion in the middle. This is what a chondrodystrophy dog looks like, where the nucleus pulposus is no longer liquidy and gelatinous. It's actually hardened and feels more like cartilage at this stage. So this is an early stage of degeneration where the liquid's lost and it's hardened. And this is what chondrodystrophic dogs have at a very young age. So while Hansen had actually looked histopathologically at discs, because we didn't necessarily know the genotype of each and every dog that he had evaluated, and we didn't know whether chondrodysplasia might be contributing to the chondrodystrophy phenotype. So a lot of the dogs, like Pekingese and Dachshunds, have both chondrodystrophy and chondrodysplasia. We chose to look at Nova Scotia duck-tolling retrievers that don't have chondrodysplasia. And we, by chance, well, let me just back up and say, maybe it's not obvious, but I want to point out that you can't look at these discs the way I've shown here in a live animal. So these are all, you know, after the dogs died that we can actually get in and look at these. And so in order to do this in tollers, we took advantage of some puppies that had died from unrelated causes, and then we're able to look at their discs histopathologically. So we had a couple of normal 10-week-old puppies, and then a 10-week-old toller puppy that had chondrodystrophy, and a 2-year-old toller that had chondrodystrophy. So if anyone wants to take a guess, there's a dog shown in each of these panels. On the left is actually the dogs without chondrodystrophy. So remember, I showed you that sort of gelatinous, watery disc that's supposed to be able to cushion, and this is what it looks like when it's cushioning. This is what it looks like in a chondrodystrophic dog, where instead of being cushiony, it's been replaced with chondrocytes and sort of turned into cartilage. So this is some work that we published in Nova Scotia Duck-Dwelling Retrievers a couple of years ago. And these results are basically identical to what Hansen saw in the chondrodystrophic dogs. Okay, so some more clarification here. IVDD stands for intervertebral disc disease, but just like chondrodystrophy and chondrodysplasia have often gotten confused in the veterinary literature, we as veterinarians, I think, have been a little loose with how we use the term IVDD. So we say disc disease when often we're talking about disc herniation. So I wanted to distinguish those for you. So disc disease would mean that it's not functioning normally, it's no longer compressible and shock absorbing, the cellular architecture is destroyed, like I just showed you in the previous slide, and the discs are now hard and composed of cartilage rather than gel. Right, so you can have increased degeneration that can occur, leading to mineralization or calcification of these discs. We don't know whether any of this causes dogs pain because they don't really complain about pain the way we do. I can tell you that in humans, if a doctor identified discs that were as abnormal as chondrodystrophic dog discs are, the person would be very much complaining and in a lot of pain. Disc herniation is where the interior of the disc actually moves from between the vertebrae. Okay, so often people say intervertebral disc disease when they mean intervertebral disc herniation. So there's a difference there. So the disc moves from being between the vertebrae. It can cause spinal cord compression, inflammation, and bleeding, and it can lead to severe pain and paralysis. And herniated discs in the neck cause paralysis of all four limbs, and herniated discs in the back cause paralysis of the hind limbs. So this is sort of a cartoon of Hansen's type 1 disc disease, where the nucleus pulposus has sort of been extruded from the fibrosis. And I think I showed you in that previous slide that what's sitting above the disc right here is the spinal cord. So when this happens, it happens acutely, abruptly, and it can actually smash into the spinal cord, causing spinal cord compression and inflammation and bleeding. Okay, so just a little summary of some of the different breeds that get chondrodystrophy and chondrodysplasia. So remember, chondrodystrophy is the one on chromosome 12, and chondrodysplasia is the FGF4 retrogene on chromosome 18. And these breeds have been sorted here based on the frequency of the chondrodystrophy mutation. So beagles, cavaliers, dachshunds, French bulldogs, spaniels, all have a really high frequency of chondrodystrophy, and these are also the breeds that have herniation risk. And chondrodysplasia occurs in some of them jointly, so dachshunds, bassets, and corgis all have both FGF4 retrogenes. And I hope that you can appreciate that their legs are a lot shorter than dogs that just have chondrodystrophy. That's mostly due to the effects of chondrodysplasia, but there are also terrier breeds that just have chondrodysplasia, and they actually have a really low incidence of disc herniation and are not seen at veterinary clinics for disc herniation. So we wanted to investigate this a little bit further by looking at a large set of dogs that that we knew had disc disease, and we did that by picking cases at our hospital that had had disc surgery. And then we looked at the breeds that were involved, what their weights were, what the radiographic evidence of disc calcification was, and what their genotypes were for the FGF4 retrogenes. So we did this using 61 different breeds, as well as a large number of mixed breed dogs that were also seen with disc herniation. And the next couple of slides are all from this paper that I'll tell you what we identified in this study. So the first thing is that having a single copy of chondrodystrophy or the FGF4 retrogene on chromosome 12 was enough to increase or decrease the age of onset of disc herniation by a couple of years. So this means that it's acting in a dominant fashion. So it only takes one copy of chondrodystrophy to alter the age with which we see presentation of disc disease to the hospital. We also identified that it acts in an additive fashion on disc calcification. So if a dog had one copy of chondrodystrophy, they were more likely to have disc calcification than if they didn't have chondrodystrophy, but if they had two copies, they were even more likely. So this is additive rather than dominant. So one copy is bad and two copies is worse. We also found that the rate of having at least one calcified disc differed significantly by breed. So Dachshunds were most likely to have calcified discs. French Bulldogs were second. Mixed breeds were actually third, and then other purebreds. We wanted to try and determine what the risk of disc surgery was in dogs with chondrodystrophy, and the largest group that we had to look at that were segregating chondrodystrophy were mixed breed dogs. So a couple of things that we determined. One is that chondrodysplasia did not seem to be increasing the risk for disc surgery, and that the risk was high in dogs with just a single copy of chondrodystrophy. So there wasn't really a significant difference in dogs that had one copy or two copies with respect to disc surgery. We also were able to look at a few breeds where there was segregation. In other words, that we had dogs in the hospital and samples that we could genotype that hadn't had disc surgery and those that had disc surgery to look at the relative risk. And so this is the risk that if the dog has a copy of chondrodystrophy, that it's likely to herniate. So it's 5 to 15 times as likely to herniate as a dog without chondrodystrophy. So while this is useful, it doesn't really tell you how often a dog with chondrodystrophy will herniate. And that's actually not been easy to determine. But one of the things that we found was a paper that looked at laboratory beagles in Japan. So the paper wasn't about disc herniation at all. It was actually about what diseases these laboratory beagles ended up having in their lifetime. And what I will say about the laboratory beagles, and I could send you a copy of the paper if you want to read it, but these were not dogs kept in cages. These were dogs that had environmental enrichment and playtime and the ability to move around and do other things. And all of their health problems for their entire lives were recorded in the study. And so beagles, by the way, are homozygous for chondrodystrophy, so they all have two copies of it, and they don't have chondrodysplasia. And in this data set, about 10% of the beagles had intervertebral disc protrusions or herniations. I talked to some beagle breeders about it, and they thought that number was low. So that's interesting, but this is what, at least in laboratory beagles, was published. Another study that we did to look more in detail at calcifications and to try and evaluate dogs that were segregating chondrodystrophy was a study that we did in Nova Scotia duck-tolling retrievers. So we did whole body CT scans in 21 Nova Scotia duck-tolling retrievers, and we divided them equally into the 3 genotype classes. So no copies of chondrodystrophy, 1 copy, and 2 copies. And this paper was just recently published, but when we look at the number of mineralized discs between the different copies, Dogs that didn't have chondrodystrophy had no mineralized discs. Dogs that had one copy of chondrodystrophy had more mineralized discs, and dogs that had two copies had even more mineralized discs. And so this was basically the same result as what we saw in the study of surgical cases at UC Davis, that one copy of chondrodystrophy increases the probability that a dog will have disc calcification or mineralization. These little stars are here because these dogs were all completely clinically normal. Their owners didn't report that they had any back problems, and they received a complete orthopedic and neurologic exam and were normal. But 3 of the dogs actually had disc herniations. And those herniations had happened sort of to the side instead of dorsally, so that they didn't hit the spinal cord, and the owners hadn't noticed any issues. So one thing is that, and I've talked to some other neurologists about this, that they do see evidence that there can be disc herniation that doesn't lead to paralysis. And so that's what we saw in this paper. The same paper also showed us that dogs with one copy of chondrodystrophy are more likely to herniate more proximally. So this is the location of the I said herniate and I meant calcified discs. The location of the calcified discs along the vertebral column. So this is cervical, so the neck area, the thoracic area, the lumbar area, and so on. And dogs with 2 copies are shown in white bars, and dogs with 1 copy of chondrodystrophy are shown in black bars. So it was interesting that the distribution along the spine was also a little bit different in dogs with 2 copies. So that may contribute to dogs with 2 copies being more likely to herniate. The most common area for herniation in chondrodystrophic dogs is sort of the thoracolumbar area right here. But we still did not see a herniation difference in the surgical dogs between 1 and 2 copies. Okay, so I bring up this calcification because a lot of people and dachshund breeders are really interested in selecting against disc calcification as a way to move towards breeding dogs that won't have herniation. But there are studies that say that there are calcifications of most, but not all discs that have herniated in dachshunds. So when they actually do histologic examination, not all of them are calcified. And there are also disc extrusions that occur in the disc that doesn't have evidence of calcification. So just because a disc is calcified doesn't mean that that disc is going to herniate. So disc herniation can occur without any evidence of calcification, and that's from this paper. All right, so let's talk about Portuguese water dogs for a little bit. So this is the results of the chondrodystrophy testing in Portuguese water dogs that was done at the veterinary genetics lab, and they shared that data with me for me to share with you. So I want to just take you through each line because they're a little bit different. So this is the number of dogs tested, the number of dogs that don't have chondrodystrophy, the number of dogs with 1 copy, and the number of dogs with 2 copies. And these are different populations. So the 636 is every dog that was tested at the VGL, whose owners allowed that information to be used for research. 328 were dogs that were relatively unrelated to each other, so that had at least 1 degree of separation between them. So the idea is that if someone identified that their dog had a copy of chondrodystrophy, they might be contacting other breeders and be more likely to have relatives tested. And so this is to sort of try and get around that to get an idea of what a true population frequency was. These are the number of dogs that were tested through the Portuguese Water Dog Club or Foundation coupon code that was used. And you can see that the percentage is less there. And then the coupon code with 1 degree of separation. So about 20%, maybe 25% of Portuguese water dogs that were tested have one copy of chondrodystrophy, and about 2 to 3% have 2 copies of chondrodystrophy. So I was curious, because I guess the information I got from the breed club is that disc herniation is not necessarily a problem in the breed, and it's a little bit difficult to sort of look that up and evaluate that. But one bit of data that I found that I thought I would share with you guys is some Agria insurance data. So in Sweden, and in parts of Europe, a lot of dogs have pet insurance, and in Sweden, most dogs are insured. Also, most dogs in Sweden are purebreds as well. And this was just one, and this insurance data is available publicly. I think that you can get to it, but this is just one way to look at the insurance data. And that is that when they looked at spinal issues versus hip dysplasia or pelvic issues, spinal issues were actually more common in Portuguese water dogs than hip dysplasia was. So red is the frequency in Portuguese water dogs, and blue is across all breeds. And I think in general, this insurance data is to be interpreted that, you know, if there's something that is higher in your breed than other breeds, then maybe that means that it's something that you should worry about or be concerned about. However, if you set that criteria for spinal problems or disc problems, like no one's ever going to be worse off than dachshunds, and so that would mean that dachshunds are the only breeds that should be evaluated that way. So I think that's a little bit less important than the fact that spinal issues, at least in this group of dogs, were more common than hip dysplasia in the breed. Okay, so I'm sure there's going to be lots of questions about that. So why might the frequency of chondrodystrophy increase? And the first reason is that it affects bone. And I mean, I'm also a dog show person, so I realize that a little bone is good and a lot of bone must be better. And over time, people could be selecting for more bone. And so I remind you what these radiographs look like, and that this dog with shorter legs also has more bone. The bone looks thicker. We were curious what different phenotypes we could identify associated with chondrodystrophy, and I was fortunate enough to spend a sabbatical in Switzerland. And one of the reasons why I chose to go to Switzerland is that there's a little known breed in Switzerland called the Schweizer Niederlachhund. And that's the only reason I got to go, is that I can say that. So the Schweizer Niederlachhund is a small Swiss hound. And the sort of breed standard phenotype is not all that important. What's more important is how well they use their noses. But I've shown 2 dogs of this breed here that have really different leg lengths. And what we knew before I went to Switzerland is that this breed segregated both chondrodystrophy and chondrodysplasia with enough frequency that maybe if we measured the dogs, we could figure out the relative contribution. So this is a little bit of a confusing chart, but what I want to show you is that chondrodystrophy is everything in orange, chondrodysplasia is everything in blue, and this is sort of 0, and then if it's to the left of 0, it means that the effect of the gene is to decrease the percentage length, and if it's to the right of 0, then the effect of the gene is to increase the percentage length. So the first thing we'll start off with at the top is that chondrodystrophy in orange actually significantly increased the skull width in these dogs. So you can imagine that having more back skull might be something that dog breeders would select for. The other thing is that height and the length of the long bones were all affected by both retrogenes, but the vast majority of the effect is due to chondrodysplasia. So the difference in a chondrodystrophic dog between 1 copy and 2 copies, the height difference is maybe 10%, which is really small if you're talking about, you know, a 20-inch dog, right? It's not a lot. Whereas for chondrodysplasia, it's a lot higher. It can be as much as 25% of the height. So it's one of the things that we use to encourage dachshund breeders to maybe consider getting rid of chondrodystrophy, because their dogs will still have short legs if they just harbored the chondrodysplasia mutation. Okay, so just sort of a summary of the different things that chondrodystrophy can do. It causes intervertebral disc degeneration, and in that case, one copy appears to equal two copies. So this would be dominant situation. It increases the risk of disc mineralization, and here it's additive. So two copies have an even greater risk than one copy, which has a greater risk than no copies. It increases the risk of disc herniation. It makes it 5 to 15 times more likely that a dog will herniate. And it decreases the age of disc herniation. So dogs with chondrodystrophy are more likely to herniate at a younger age than a dog without chondrodystrophy. And then I guess sort of in the green or plus category is that it shortens legs by 5 to 10%. And it does that in an additive fashion. So two copies dogs will have even shorter legs than a dog with one copy. I just want to pause here and say that this drove the Nova Scotia duck-tolling retriever people absolutely crazy, and they would show me many examples of tollers with two copies that were 20 inches tall, and how could that possibly be? And I would explain that if they didn't have two copies, they would have been 24 inches tall. But you know, the breed standard is something we just completely made up. This is the genetics. It's not the breed standard. So it doesn't really care what the breed standard is or how tall they're supposed to be. But on average, this is what it does and how much it would decrease it. Chondrodystrophy also increases skull width, and this looks like it's pretty dominant, and it's about a 20% increase, which is about the same as the difference between a bitch and a dog, just to give you an idea. It also may increase the ear width at the tip of the ear. Dr. Ostrander's group also found that something in this area of the genome increases ear width, but they don't think it's chondrodystrophy, and we disagree on that. I've looked at lots and lots of puppies that segregate chondrodystrophy in tollers and in other breeds, and they definitely have rounded ear tips. But okay, so the mode of inheritance is dominant, and this is a problem. And you know, it's really easy to handle and deal with recessive disorders because animals that have one copy of a mutation are completely normal, and you can breed them and you can produce them and they don't have disease. But that's a problem for chondrodystrophy. So I think one of the questions that I was asked beforehand was, you know, what is the mode of inheritance of chondrodystrophy? And it really depends on which phenotype you're referring to. So IVDD, standing for intervertebral disc disease, which predisposes dogs to herniation. Chondrodystrophy causes IVDD, but that only predisposes to disc herniation. It doesn't necessarily guarantee that that dog is going to go on to herniate. And there are lots and lots of dogs with chondrodystrophy that do not have disc herniation. Their discs are all abnormal, but they haven't randomly slammed into the spinal cord and caused them to have pain or paralysis. We don't know of any protective alleles, but herniation rates do vary between breeds. For example, dachshunds have the highest estimates of herniation, and they're much higher than beagles. And even different varieties of dachshunds have different herniation rates. Every dog that has ever been evaluated with chondrodystrophy that's had histopathic evaluation has abnormal discs. But that's not to say that they all do, because it's not something that's easy to evaluate. And you have to do it in young dogs, because all dogs, when they get older, will have changes in the disc. And so the point here is that this is happening prematurely in dogs with chondrodystrophy, so you would have to look at younger dogs to see that. I'm going to get to my breeding recommendations in a minute, I promise. So I thought you might be interested in this data. So inbreeding in the Portuguese water dog. So based on genotype, not pedigree, but genotype, Dr. Boyko's group had estimated that Porties have an average inbreeding coefficient of 0.22. In a paper that we published, it was 0.23. This is from that paper, and it's a diagram that shows what the average inbreeding coefficient is across a large number of breeds of dogs. So the average here was 0.25, and 0.25 a little bit higher than what Porties were shown to have. So they're, you know, slightly better than the average, but all purebred dogs, or the vast majority of purebred dogs, are highly inbred. And so people get really worried about, you know, if we select strongly against this, we're going to change or alter our inbreeding or increase our inbreeding. And I think that there are ways to manage chondrodystrophy without doing that. So if you avoid breeding heterozygotes to each other and instead breed dogs with chondrodystrophy to dogs without it, and then replace the chondrodystrophy dogs with normal dogs, you're not necessarily losing any dogs from breeding. But the question here is, how do you sell puppies with the knowledge of their chondrodystrophy status? And that's really a challenge. I think it's particularly a challenge where this breed is kind of sitting with the rate of dogs with one copy of chondrodystrophy. I mean, it's relatively low. I know 25% seems really high, but compared to tollers, it's lower. So in tollers, the percentage of normal dogs is basically about 25%. So half the breed has one copy of chondrodystrophy, 25% have two copies, and 25% have none. So that makes it a lot harder to choose to breed away from it, because you really are affecting the gene pool. I also suspect that maybe the overall rate of chondrodystrophy in Portuguese water dogs is a little bit lower than those numbers that we saw, that's probably due to people who were, you know, contacting each other and talking about testing relatives. So how do you sell the puppies? I think that what we've done in tollers is to tell people that the breed is trying to move away from this. I will tell you, I think Porties have 2 mutation tests, maybe 3, right? So you have the early dilated cardiomyopathy, storage disease, and PRCD. I can't see you guys to let you answer this question, but I will just tell you that tollers have 16 mutation tests. So it is a lot harder in tollers to take into account all of those different mutation tests and try and breed away from chondrodystrophy. But when you're in a situation where you don't have very many mutation tests, I think that it should be a little bit easier. And so I completely stole this from the awesome graphics that were available on the Foundation website for recessive diseases, but I'm sort of co-opting it to tell you about dominant diseases. So this is what I'm promoting that you do. So I think everyone individually would have to make a decision about whether they wanted to breed a dog with chondrodystrophy or not. But obviously there's a lot of other things that went into creating those dogs, and there's other traits that you might really like about those dogs. So I'm assuming that there would be motivation for breeding a dog that had chondrodystrophy. And if you bred one that had chondrodystrophy to a dog that didn't, you'd get 50% of the puppies with chondrodystrophy and 50% without. So what I suggest is that that litter be tested, the puppies without chondrodystrophy be the ones that go into breeding homes, and that these not go into breeding homes. And that way you're not losing this dog from the gene pool, you're still breeding it, but you're moving on, getting rid of the mutation. And just to point out the other scenarios, if you had a dog that has 2 copies of chondrodystrophy and you breed it to a dog with no copies of chondrodystrophy, all of the puppies will have chondrodystrophy and it's dominant. So all of these puppies would be at risk of herniation. And if you breed 2 heterozygotes to each other, so dogs that have 1 copy of chondrodystrophy, 75% of the puppies would have chondrodystrophy. Okay, and then we have the question slide. So I am happy to take questions. I am going to put this thing away, and maybe I'll have to refer back to it later. But so, questions?
Oh, thank you, Dr. Bannasch. That was very interesting. I do have some questions from the list that I had compiled earlier, and they reflect the questions that I've read on various Facebook groups over the last couple of weeks. A lot of people are concerned about how chondrodystrophy looks different on X-ray compared to a dog that just has arthritis of the back. Can you do a plain film of the back and make the diagnosis with just plain X-rays?
So not really. What you can see with plain X-rays, if your radiographer is very talented and skilled, is disc calcification. But you absolutely can't see anything that would indicate that the disc has degenerated. We used CT scans to do the disc calcification that we did in the tollers, because that is really sensitive to calcification. Usually from a diagnostic standpoint, if a dog has potentially herniated or showing clinical signs that it's herniated, so it has severe neurologic deficits, an MRI will be done. And when you do an MRI, you can actually visualize the abnormal nucleus pulposus and see that it's herniated and figure out where surgery needs to be done. But yeah, it's not easy. Now, there are dachshund breeders, and in Europe, they are promoting radiographic screening for disc calcification in dachshunds. But those are dogs that mostly have 2 copies of chondrodystrophy and lots of calcifications, but you can't really use that generally in any breed. And one thing that they do in the dachshunds is they set the age of dogs that they are going to radiographically screen. So they screen them all at, I think it's 4 years, might be 2 years, and then decide based on the results of that whether they should be bred or not.
And how successful do you think that is going to be in their breeding programs?
Well, yeah, there's a lot of controversy about that online. So the problem is that, I mean, so chondrodystrophy does actually segregate in wire-haired standard dachshunds in Europe. So that's why it was actually, disc calcifications were actually mapped to chondrodystrophy because it segregates. So when it segregates, then you can select against chondrodystrophy. But in the breeds where it's fixed, I'm not convinced that having more or less calcifications are going to make a big difference. And they have been employing this selection scheme in Finland for a number of years, and they really haven't made much difference at all. So
many people have expressed, as you already dealt with to some degree, that the Portuguese water dogs don't seem to have a lot of back problems. And they're wondering whether or not this FGF4 expresses in the Portuguese water dog. Has it been measured specifically in the Portuguese water dog? And if you were going to measure FGF4 in the Portuguese water dog, how would you do that?
Yeah. So no, I mean, honestly, other than the fact that this other paper was published saying that it was segregating in Portuguese water dogs, certainly are not a breed that, you know, we're aware of that have a higher rate of herniation. And I mean, I understand that there have been some Porties that have herniated recently, which is why there's a lot of interest in it. But as far as I know, I mean, I looked for that agri insurance data to share with you guys because, you know, it's not something that was particularly on our radar. I went into our hospital database to see if there were any Porties that had herniated, and there weren't. Mind you, there aren't really any tollers that have herniated in our database either, because we don't see very many of them. I do want you to know, anyone who comes to UC Davis, that I have successfully changed the breed name in our hospital from Portuguese Water Spaniel to Portuguese Water Dog, which just really irritates me. But and your question was, how could we measure FGF4? So you mean the expression levels? The way
you measure it in duck-toling retrievers? And is that a practical way? Can we donate some dogs to be measured?
Yeah.
Is it a blood test?
So we didn't measure FGF4 levels in tollers. We actually measured them when I showed you the disc measurements and said it was 20-fold higher. That was, those discs were carefully surgically extracted from tails that had been docked. And so we instead of having to take a puppy, we took a tail that came from a puppy. I will not euthanize a dog for research. So it's not an option. Now, if you had puppies that died from other causes that had chondrodystrophy, you could, I don't want to speak for our pathologists whether they would do it, but you could have histopathologic analysis done on their discs to see if they looked chondrodystrophic or not. So that's definitely a possibility. And I know that the Breed Club has a long, rich history of necropsies on older dogs, right, with the Georgie Project, but this would have to be on younger dogs.
I appreciate that. I really wondered if it was just a blood test that you did, and I had no idea it was actually tissue analysis that you're doing to measure these levels, which is a whole different, we don't dock our tails either.
Although,
I don't think we're going to start either.
No, I'd have to double check. I can't remember. It's possible that maybe there's expression in testes, which would mean that we could maybe look at neuter. I don't remember. I mean, that would only happen with chondrodystrophy. So FGF4, parental FGF4 is absolutely off in adult dogs, but in chondrodystrophy dogs, there's some remnant of expression in the discs and in the vertebral bodies. And I can't remember if maybe there might have been some low-level expression in the testes, but I could look into that and let you guys know.
I was wondering.
Oh, it says to turn my camera back on. I'm so sorry. I didn't even know my camera was off.
Yes, we can't see you.
I have to apologize to you guys.
Thanks.
Like, normally I use Zoom and I have no problems doing all sorts of wonderful things. You even missed all the fun, fancy things that were going to fly in on my slides. Apologies.
I have a question about Portuguese water dogs with CDDY who are pregnant. Are they at increased risk of rupture of their back during pregnancy? Is there any softening of the discs like occurs in pregnant women where there's softening in the pelvic joints to facilitate delivery? Does anything like that happen in the back?
Yeah, that's a really good question. I will say that in tollers, we definitely have had, I can think of 5 or 6 examples where toller bitches post-whelp herniated. And that seems oddly high, and it's not uncommon. So I do wonder if either they're, jumping when they shouldn't jump, jumping in and out of a whelping box, or you could imagine maybe someone has an X pen up and they're jumping in and out of an X pen and that's why they herniated, or whether there is a change post-delivery. And I don't know the answer to that, but we have seen that in tollers. I have not really heard or seen that in dachshunds, but I'm not sure that I would know.
Another question about the concerns about losing positive genetic material by breeding away from CDDY. Does it work the same way when you start breeding away from an allele that is extra as when you're trying to breed away from a mutation that is a change in a gene, as far as protecting the gene pool? In other words, are there genes that are likely to lose as a result of breeding away from CDDY?
It's no different than any selection that you might employ. I mean, theoretically, if you breed away from CDDY, there might be other alleles of other genes nearby that, are not going to be there. I mean, in your case, it's a really low number that have CDDY. So, I assume that the rest of the copies of chromosome 12, that there's plenty of those. But it's not different than any other locus that you might employ strong selection for. I mean, I put up the inbreeding because I think we've sort of, I'm not sure that I ever did this, but I'll count myself in the group of geneticists that we kind of did a disservice to people. So, and I'll use tollers as an example. The toller breeders were told, be really careful, don't select against chondrodystrophy, oh my gosh, PRCD, because if you select against it too heavily when you have a high allele frequency, then you're going to get other things in the breed. And so people took that to heart and really continued breeding carriers. And then the breed club had the unfortune of having 2 geneticists decide that they love the breed, and now we have 16 mutation tests, and there are people who cannot dig their way out. But I mean, as a toller breeder, the first thing I did, there's a mutation test that came out, I cleared my dogs from that and moved on. Another mutation test came out, I cleared my dogs from that and moved on. And people were afraid to do that because of what it might do to the breed. And I put the inbreeding to show you they're already highly inbred. Like, you know, I'm not sure that you can, we didn't know that at the time when these recommendations were being made. We didn't know how inbred purebred dog breeds are, but now that we do, I'm not so sure that that's really relevant.
Thank you. If you have a dog with CDDY, is there any kind of restrictions you would put on the dogs, or any kind of sports that you would suggest that the owners not participate in?
Yeah, I mean, there are plenty of chondrodystrophy dogs that compete in agility and dock diving and do all of those things. But one thing from dachshund data we know is that if dogs are kept lean and in good shape, they're less likely to herniate. There's some really odd dachshund data that dachshunds that had to climb stairs daily were less likely to herniate. And the thought was that they're in better shape. And so because they're in better shape, they're less likely to herniate. I personally would not want to do agility with a dog with chondrodystrophy, but people do. And some of them herniate. But I see lots of questions in the chat. I don't know if we should, what about water work? Yeah. Well, again, I mean, swimming, yeah, leaping off the boat, maybe not so much, but swimming obviously would be great, right? It would keep the dog in really good shape and keep them lean. And I think that all of those things are really positive. And if you know, I think you have to think about keeping your dog that you have happy, right? Like if you have a dog with chondrodystrophy and that dog enjoys water work and that dog is already an agility dog or whatever, I'm not sure that I would like make them have miserable lives because of it. I don't think that that's necessarily fair to the dog. But would I sell or buy a puppy with chondrodystrophy to do agility or water work? Well, I would be less inclined to do that. Do you want me to answer any of the questions in here? Because some...
If you see some that are intriguing. I was wondering if you...
Let me scroll back up. Now stop posting them, people, because there's a lot.
I can't see the chat. Are there any supplements that you recommend as preventatives for dogs that have CDDY?
No. I mean, no.
No is a good answer. I mean,
I'm sorry, keep them lean, and I'm not a nutritionist. So do I have actual herniation rates for Porties with 0, 1, or 2 copies? No, I don't. And you know, it would be Paula. Sorry, Paula Henthorn's on here. Hi, Paula. Is there any data about the effects of conditioning on rate of herniation in dogs genetically at risk? Can you protect a dog? Yeah. So like I said, I mean, I think actually keeping them lean and in good shape and, you know, walking, not jumping, etc., is all really good. The history that we get with type 1 herniation is often like dog leapt in the air after a squirrel, dog was playing with another dog and that dog ran into him, and then they herniated, right? Normal dogs don't herniate from those types of things happening. I mean, normal dogs don't herniate even if they get hit by a car, right? So it's because the discs are abnormal that they have that sort of propensity to herniate. But I think keeping them in good shape and in good body condition is, you know, it's been shown in dachshunds, and there's no reason to think that that wouldn't be, you know, a good idea. So Is there an association between CDDY and shorter rib cage and or long loin? I'm laughing because I tell my students, my vet students all the time, dachshunds are not long backed. We don't know anything that can make the back longer. They have short legs. They don't have long backs. That being said, there's maybe a squidge of back length that'll come out from these CT studies on tollers. But I mean, it's minuscule. It's like 1% difference. So I don't think it's something that, you know, even we as dog breeders could perceive. So the question is,
could you comment on the various labs that do CDDY testing? Are they all the same? Are they doing the same test, or are they doing something different from one lab to another?
Okay, so there's one question that says, if Wisdom Health testing shows clear for CDDY, why does UC Davis show one copy? So Wisdom Health is not running CDDY yet, as far as I'm aware. Maybe they started, but I don't think so. Embark runs a linked marker test for CDDY, and we have definitely found numerous examples where it's not accurate. And I can tell you, there is not a SNP marker that can be used as a substitute for CDDY. There is for chondrodysplasia, but there isn't for chondrodystrophy. So I think you have to be careful with a linked marker test. And when it comes up positive, my advice is that they test with a lab that's actually running the test for the gene insertion and not a linked marker. But I don't believe Wisdom is doing CDDY testing yet.
You're correct.
They want to, they are ready to, but they're waiting.
I think Gensol is doing a CDDY specific test, just as veterinary genetic labs. Do you know of any other labs that do it the way you do it?
So I don't necessarily know what testing methods every lab uses. So I can't, I mean, they don't publish that information, so it's hard to say. So I can't really answer that. Sorry.
Okay.
I mean,
but if your dog has been tested at UC Davis, then we can be sure that the test is accurate for CDDY.
Yeah. Yeah.
Thank you.
And I'm sure there are other labs that are accurate too. I just don't necessarily know how they're testing. Okay, so someone wrote, yeah, Wisdom tests for chromosome 18, which is chondrodysplasia, not chondrodystrophy, and that I believe. Another question is, so I understand you are recommending not to breed 2 copies of chondrodystrophy. I would personally have a hard time doing that. If you as a breeder can breed a dog with 2 copies of chondrodystrophy and tell the puppy buyers that all of the puppies are at risk for disc herniation, no matter how low it is. I mean, we didn't talk about this. Disc herniation is, number 1, probably the most painful condition that dogs can have. Number 2, it costs about $12,000 between diagnostics and surgery. The VMTH at UC Davis does surgery on 1 disc dog every day, and we make about $5 million a year from it. So please keep breeding it. I mean, I'm joking. I don't want to see another dog with a zipper back, that's what I call them, in horrible pain. And it's a horribly painful condition. And of course, not all dogs with chondrodystrophy will herniate, but the ones that do are in horrible pain. And I can't get behind that. Anyway.
Can you easily distinguish between an old dog with just degenerative changes from aging from a dog with CDDY?
Not necessarily in an old dog. We were just, neurologists and I here were just talking about that. Like, should we do some sort of a longitudinal study using MRI to sort of look at when the discs sort of lose their kind of normal watery structure, proteoglycan structure, which you can see on MRI, but no one's really done that. There are plenty of studies that have looked at chondrodystrophic and non-chondrodystrophic dog breeds, but the breeds were wrong, and even some of the dogs were segregating, right, and they didn't know about it. So those studies definitely show a difference, and the breeds are wrong. So I imagine that it will only get a lot better when they actually have the breeds correct. But anyway, So
Are there behavior changes associated with CDDY in dogs? You're aware of any
that's an
behavior
interesting
changes?
question. Yeah, I wonder if a dog herniates and it doesn't have neurologic deficits, so the owner doesn't know if that doesn't cause pain and maybe could cause behavior changes. I think it probably depends on the dog a little bit. So someone in the chat has pulled something that Dr. Dickinson and I wrote out of a paper asking about RSPO2. So RSPO2 is a gene that causes beardedness, facial hair in dogs. And it is in the same pathway as Wnt, which may be important for disc development. And we were speculating in the discussion that, you know, maybe there could be differences in different coat types. Maybe the reason why wire-haired dachshunds are less likely to herniate and long-haired dachshunds are more likely to herniate has to do with the fact that their coat type mutations are in this pathway. And obviously Porties would have the RSPO2 gene mutation. Yeah, I mean, it was speculation, so we don't know. Poodles also are bearded, and when poodles were more popular, they were, you know, high up on the list of dogs that herniate, and they definitely herniate. So it's not completely protective if it gives any sort of ounce of protection. But, other questions? Sorry, I think I answered that one. Question of whether Gensol is accurate. I have no idea. So there's a question, do I recommend neutering a dog with 2 copies? I mean, neutering doesn't have anything to do with breeding. So I wouldn't breed a dog with 2 copies, but you could breed a dog with 2 copies and take 2 generations to get to a normal dog if you wanted to. Neutering dogs does tend to make them less energetic. So if it meant that the dog was going to be overweight, that might not be in its best interest. If a dog has a copy of CDDY and loses use of its back legs, is it a disc herniation or can it just be from pressure on the spinal column? I mean, if a dog has CDDY and loses use of its back legs, it needs to see a neurologist and have diagnostic imaging done. I mean, we wouldn't say anything based just on its CDDY status. So, what percentage of Portuguese water dogs with 1 copy will actually be affected? I do not know. I do not know. Maybe it's worth it to try and you know, take a poll and do a study on that. Whoa, sorry, I'm trying to read the chat, but it's moving.
There's a question here about a minimum age to test for CDDY. You can test at birth if you want. I mean, you can test a puppy at any age. It's not going to change. Wire dogs with 2 copies of CDDY and over 10, active in agility and not showing any symptoms. So CDDY is not always expressed. Okay, so I tried to explain this. Dogs with CDDY have abnormal discs. That doesn't, the herniation event is this sort of catastrophic event that can happen if a dog has abnormal discs, but it doesn't mean that it always happens. But their discs are still abnormal. Their discs are still not cushioning as well. And we don't and can't ask dogs if they have back pain, but I certainly in my experience, because I've lived with a lot of chondrodystrophy dogs, they have back pain. And, you know, they like it when they get painkillers and they don't want to do things when their backs hurt. So
yeah, I mean, it's all anecdotal, but yeah, my agility dog, Master Hunter, who retrieved the kickoff tee for the UC Davis Aggies for 44 games and 11 seasons, I put a special new harness that we ordered with UC Davis all over it on him, and it hurt his back. It like pressed on the thoracolumbar region and caused him back pain. He has chondrodystrophy. I don't know. Okay. So, right. So just again, disc disease is not the same as disc herniation. So I know everyone would love a DNA test for disc herniation, but I don't think that's ever going to happen. And when we've tried to map disc herniation, like with endoxins, you just get chondrodystrophy. You don't get anything else. So
some of the labs report CDDY as being variably penetrant. Can you discuss what about CDDY is variably penetrant? Yeah.
So modes of inheritance kind of depend on where you're defining the phenotype. So, and I had a slide on this, but intervertebral disc disease, IBDD, disc disease, it's 100% penetrant as far as we know. I mean, Hansen, Brond, our lab didn't find any chondrodystrophy dogs that didn't have abnormal discs. And so histopathologically, all of those discs are abnormal. And they're abnormal at a young age or a younger age than they would be normally. So it's 100% penetrant if you define it for disc abnormality. If you want to define it as disc herniation, which it's not a test for, but if you want to define it as disc herniation, then it would be incomplete penetrance, because there are plenty of dogs that have chondrodystrophy that never go on to herniate. That doesn't mean that it's something that you want to have around in your breed if there is a risk of herniation and you have a relatively modest allele frequency. So why do you think the incidence of the gene is low? Because we possibly selected away from it because of shorter-legged puppies. Yeah, that's what I think. I mean, I think that my guess is I read your breed standard and talked to my Porty friends, and, you know, slightly longer than tall. I mean, it's almost like a given that when we see slightly longer than tall, it seems like chondrodystrophy is in the breed at low levels. But I think it's the fact that it's slightly sort of saves you, in that if they get really short legs, or if they get crooked, because you're definitely supposed to have straight legs, they get too much bone, they're too coarse, and you're probably selecting against that. And so there's just a low level allele frequency, probably because of the benefit that it would have on adding bone, maybe the back skull, I don't know. I will tell you that a friend of mine who actually breeds Danish Swedish farm dogs calls it the pretty gene, which just kills me. But if you set up, you know, a dog with chondrodystrophy, we don't even have to use tollers, but we can even use Porties. A dog without chondrodystrophy and a dog with chondrodystrophy, one copy next to each other, and showed them to a breed judge, they pick the dog with chondrodystrophy. Like it makes judges select for it. And so you ask how it got spread in the breed. I mean, it's probably because judges selected for it, or people selected for it because of its influence on bone. But, sorry, I'm trying to read this. OFA designates PWDs with at least one gene copy as CAR, which sounds, at least to me, like simple recessive nomenclature and potentially confusing. Yeah, so I tried really hard not to use the term carrier, because from a true genetic standpoint, carrier means that they're completely normal. And in this case, it's dominant, so there are no carriers. And so when I say they have chondrodystrophy or they're affected, I'm talking about dogs with either one copy or two copies. In my mind, they're equivalent. So yeah, it's a matter of them probably just not having the ability to call it a heterozygote, which is the technical term for it. The question is, what technique does UC Davis use to test for chondrodystrophy? I don't know. Yeah, I don't know. I guess I could ask them. They don't use the same one that I do, but they use something. Do we have data on what percentage of dogs with chondrodystrophy actually herniate? Not in Porties, and I gave you some data on that. It varies by breed. I think probably the thing that is most similar to compare to would be the Beagles, because they're homozygous for chondrodystrophy, and about 10% of them have a herniation event that led to clinical illness in their lifetime. And I also showed you that in Tollers, there were dogs that were asymptomatic that had herniated. So there's more herniation going on that's not necessarily resulting in clinical signs of neurologic dysfunction. Could chiropractic services, acupuncture help keep a dog with 2 copies to help keep compression minimal? I don't know. I don't know. More questions?
Seem to be winding. I think I've asked the question.
Paula asked, is the CDDY allele in additional breeds? Yes. So CDDY is the most popular mutation across all dog breeds, apparently. It occurs in 12% of dogs tested by Wisdom. So it's in many, 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 many dog breeds. And it segregates in many dog breeds. More than DM, yes, sadly, #1. I was really excited because hyperuricosiuria, which is a mutation that I identified, was #4 a couple years ago. And I was like, yeah, my mutation is the 4th most common. Yeah, but chondrodystrophy beats that. So yeah, it's in the most number of breeds.
Some people have asked about the degenerative myelopathy and CDDY and how they're different.
Yeah, so I think that, and it's been pointed out by a few neurologists too, that you really need to do diagnostic testing, because the beginning of degenerative myelopathy and chondrodystrophy could look similar. Normally, chondrodystrophy is acute and painful, but there could also be, or intervertebral disc herniation is acute and painful. But there can also be kind of chronic old age changes that might look similar to degenerative myelopathy. Degenerative myelopathy is normally non-painful, so that's one thing that would distinguish them. But
can you describe how one should manage a dog if they suspect they've just ruptured a disc?
Yeah, well, definitely, not letting it move anymore or move around. So immobilizing it in a crate and going to a neurologist as soon as possible to determine how bad the disc rupture is. So if a dog loses deep pain sensation, it's not considered as good of a surgical candidate. The faster the disc is decompressed in surgery, the better. But there are definitely other ways to manage dogs with disc herniation too. So it's not, surgery isn't always the answer, but it's something that is normally handled by, I should clarify this, neurologists or surgeons. So at our hospital, our neurologists are neurosurgeons. Some neurologists aren't neurosurgeons, and surgeons would actually be the one to handle it. But this is something that's seen really commonly in emergency medicine. So if it's after hours, you're going to an emergency vet and getting some relief for the dog. I think the main thing is that it is incredibly painful, and they should be put on pain meds as soon as possible. Just help them.
How about the use of steroids?
Yeah, so I'm not a neurologist, I'm a geneticist, but yeah, people can treat them with steroids and pain meds. And in fact, it's my understanding that the Beagle people kind of put them on cage rest and give them steroids and pain meds until they kind of recover on their own. But yeah, not a neurologist. Someone needs a basic back Can stem cell therapy help reverse disc degeneration? Certainly, as you can imagine, if anyone has back pain in the audience, which I'm sure you all do, because I do, there's a lot of interest in animal models for human disc disease and back pain, although the dog chondrodystrophy condition is actually a lot worse than what we see in people. People don't herniate nearly as often as dogs do. But there are people looking at stem cell therapy for dogs. I haven't really seen anything that reverses disc degeneration, but they're working on it.
Are there any questions that we forgot to ask you that you would like to answer?
Well, I would really like to know, I mean, how often have porties herniated? I mean, is it a problem? Is it not a problem?
If you were to design a study to evaluate CDDY in the Portuguese water dog, how would you suggest that be done?
Well, so I was just reading, we had a PWD herniate a disc prior to this test, but her UC Davis test is NN. That would be strange, but could happen. There are other, so one thing that is a point of confusion, I think, with breeders is that chondrodystrophy causes premature degeneration of the discs, but as dogs get older, any dog can have disc degeneration, and there's different types of disc disease in dogs. So I didn't really outline those in this talk because we were talking about chondrodystrophy, but there's Hansen's type 2 disc disease, which is usually a single sort of chronic disc. It's actually more similar to what people get, where you just have like one disc that's kind of a mess. It's got osteoarthritis. It's painful. Surgery is not necessarily recommended for those cases. It depends. It's a case-by-case basis. So it's a really different kind of presentation. But just because a dog doesn't have chondrodystrophy doesn't mean that it's not going to ever have a back problem. There are other back problems in dogs. Dogs can get spinal tumors. They can get fibrocartilaginous embolism. They can get other things. It's just that they are less likely to have these sort of abnormal discs that are predisposed to acute herniation at an earlier age.
Do you think that doing a prospective study with age-matched and sex-matched control group of CDD positive and CDD negative dogs would be the way to go forward to study this in the Portuguese water dog?
Yeah, sure. I mean, it's just going to take, 15 years, right, to follow a cohort of dogs and see what happened to them. But it's possible. The other thing you could do is, if you wanted to know if the discs were abnormal in porties, if puppies die of other causes, you could do the histopathologic analysis like we did on the 10-week-old Toller puppies and genotype the puppies to compare dogs without chondrodystrophy to dogs with chondrodystrophy. Yeah. CT scans are a little bit hard because the way we analyzed that CT data was really not something necessarily routinely done by radiologists. It was done on a research basis, so I'm not sure if that would quite translate. Are there any breeds with CDDY dogs that never herniate due to CDDY? Not that I'm aware of. Like, every breed that we have found it in, or that has approached us because they've had individuals that appear to have Hansen's type 1 disc disease, we've found the mutation in. So it's a lot of breeds, I mean, that we've worked with. So Coton de Tuilers, Boykins, Chesapeake Bay Retrievers, Tollers. I mean, these are odd, sort of non-typical breeds. I think, what else? Yeah.
You mentioned to me, a private conversation that 2 copies of CDDY could be associated with changes in the top line of the dog.
Yeah, yeah. if I don't think we want to go back to my slides, but yeah, so in tollers with 2 copies, we definitely get sort of this kind of reverse sloping top line. So there's sort of a dip behind the shoulders, and sometimes it's level if you really pull up their heads really tall or stretch out their hind legs, but it can be not level. Yeah, it wasn't something I measured in those Swiss dogs. So it's a confirmation thing that I'm not even sure if I can get our orthopedic surgeon to see. The other thing that I've seen in tollers is that dogs with chondrodystrophy stand wider in the front. And we will be looking at the humerus in those CT scans to see how the humeral geometry is different. So that paper will be coming out at some point. But oh, the question was about poodles. Is it in standards? It's incredibly rarely in standards, like almost never. It's predominantly in toys and miniatures. We've had a few standards that actually had, the reason why we tested them is they had really crooked front legs, sort of reminiscent of those tollers with 2 copies. And it turns out that they had, I don't even think they had 2 copies of chondrodystrophy, but they had 1 copy of chondrodystrophy. By the way, I didn't really come full circle here and mention that we still don't know why those tollers have really crooked front legs, because while all of the tollers with crooked front legs have 2 copies of chondrodystrophy, there are plenty of tollers with 2 copies of chondrodystrophy that just have short but straight legs. So we still don't know why they looked so valgus. But would I consider doing research specific to PWDs? Sure, you'd have to convince our orthopedic surgeon maybe to do it, or one of our neurologists, but yeah, I mean, I think it would be interesting to do it in another breed, and another breed that's sort of atypical. That might help convince people. I am on a crusade to eliminate this disease, and I guess this is sort of a personal thing. Like, I've developed, I think you said 16 tests, Cheryl, but it's a lot more than that. I think I've up to like 22 tests. And the reason I developed tests is because I want to relieve pain and suffering. And this disease causes a lot of pain and suffering. And it just kind of blows me away that people want to keep it around because maybe it's pretty. Like, that's just as unacceptable to me. And I feel like I should, have a video of a dog that's herniated, screaming in pain, so everyone could sort of share that and think about what that might mean for that dog and how much pain it was in and for how long. And the last thing is, it's expensive. It's $12,000. And if you have insurance, believe me, they are not covering $12,000 worth. So people are making decisions to euthanize dogs because they can't afford to treat this disease. And that also makes me really sad.
That's not exactly a positive note, but I think
I'm sorry.
We're coming to the end of our time with you.
We could do something more upbeat and positive.
Yeah, well, I think that was a real downer, but we do really appreciate your expertise and you spending this time with us this evening to answer our questions. Maybe we'll come back to you and circle around in a year or two when we have more data on our dogs and see if you have more things to add about our breed.
There's one question. Tell us how we are lucky our incidence is low. Yes, you're really lucky. Your incidence is low. You have very few homozygotes, probably because you phenotypically don't want to breed heterozygotes to each other because they probably have too much bone. So that's good. And I guarantee you can make beautiful Portuguese water dogs without this mutation. That's my life.
That's very good. Thank you very much, Dr. Bannasch, for taking the time to be with us this evening. We really appreciate your time.
Okay. All right. Thank you.
Okay. Good night.
Good night. Let's see. How do I get off of here?